All right. So here's the ovary ovulation curve, oocyte comes out here. So they're kind of large, uterine tube. Here's that infundibular multifimbriae on the edges. So where we are, where fertilization occurs, is usually about the first third of this uterine tube. So the sperm, made their way down the uterine tube, use the flagellum, reach the egg, they fertilize it. And what they're showing you here's the oocyte, and that little white thing around it, I believe, is representing the fertilization membrane. That's going to be making its way through the uterine tube. It takes it a few days. It doesn't move very quickly. That embryo eventually has to implant here in the main body of the uterus. So early development, the embryo isn't attached to anything. It's just slowly drifting through the uterine tube. So what we're going to do is start at the point of fertilization. So the fertilized egg, generally called the zygote. And then we'll just take that zygote through some of the developmental stages until basically the first organ systems start to form. Then I cut it off. So shortly after implantation, the embryo will implant. Once you get a nervous system beginning to form, I'll stop. So we only go a few days into development, really. That's kind of what's familiar. So the zygote, after fertilization, is called the zygote. And what we're looking at is basically descriptive embryology. This is the early stuff where they watch the embryos and they just describe different changes they saw. That's really all descriptive embryology is. We're not looking at other chemical controls and signals, which is some kind of the details and the nitty-gritty you're working on now. But the first thing you would notice, in a human, there's a bit of a delay. It's nearly a day while the zygote kind of rearranges the pronuclei from the sperm and the egg. They kind of recombine all those chromosomes. So a bit of a delay. After about 24 hours, though, what you would notice is something called cleavage. Cleavage is, again, right after fertilization. This is the first thing you notice. All it is rapid mitotic division. And of course, mitosis occurs from this point until you're dead, even well after you're dead. So mitosis is always going on. That's division where it's just direct or identical copies of the cells are produced over multiple generations. What sets cleavage apart from other types of mitosis is there's little to no growth in the embryo, the organism. Normally, if you remember the cell cycle, you had the G1, which is normal growth and development. Then you had the S phase of the cell cycle, that's DNA replication. Then the G2, kind of prep for mitosis, and then you go through the mitotic division. Basically, in cleavage, there is no G1 to speak of. It's very rapidly cycling. In some species, these mitotic divisions occur about every 20 minutes, so it's very rapid. And all that's happening, you're taking a cell, and it'll undergo mitosis. You'll have 2 cells. Those cells will divide. You have 4 cells. Those cells divide. Something like that. 8 cells. It's just rapid mitotic divisions. What you'll notice is the whole organism doesn't change size. In fact, let's switch to a different view here. If you look, this is the first cleavage division. Of course, this isn't a human, it's a ceratid. But what you're seeing is a cell here, and the cleavage row isn't quite finished, but it's dividing it in 2 right there. But what you notice, see that clear envelope around it? That is a fertilization envelope. That's fairly rigid. That is, it doesn't expand, it's not elastic. So as this thing divides, if you're watching it, you can see it go through several cleavage divisions. And this is the same thing here. This might have 8 or more cells in it. I can't quite count them all here. But even though you've got more cells, you still see that fertilization envelope. The embryo doesn't get any bigger. That kind of uses the gauge for how big it is. So the thing's not growing. What you're doing is you're increasing sort of the ratio of genetic material in cell membrane versus cytoplasm. When you start off with one big cell with one nucleus, you're going to end up with 8, 16 smaller cells, each with their own nucleus. So you're making these smaller cells out of a bigger cell. You're not getting any growth at this point. Mention one other thing, tubal pregnancy. Now what happens, an embryo when it implants, it's going to try and make a placenta. We talked about ectopic pregnancies, I think, early on. But normally that doesn't happen. Now, one of the ectopic pregnancies, tubal pregnancies, sometimes the embryo will plant in the uterine tube. That cannot accommodate the growing embryo fetus, so the pregnancies aren't successful. What usually prevents that is this fertilization membrane. As long as that's there, it's kind of this resistant membrane around the embryo, it won't implant. So for a tubal pregnancy to occur, that fertilization membrane has to have gotten damaged. If not, it's going to keep on going. All right, in humans, there are lots of different types of cleavage. We don't go over them all in here because we're mostly worried about humans. But the way I've drawn it, it's nice and even. You have 1 cell, and 2, then 4, then 8, then 16. Placental mammals tend to have asynchronous division or cleavage, but that means you won't always have an even number of cells. You know, you can get 1 cell here, another cell here. This one might divide first. Before that one, they're not synchronized, so you got 3 cells, you got 5. So you can't have odd numbers of cells. So cleavage will occur until you end up with a solid ball of cells. Put some diagrams on. Same size as the original embryo, the original zygote. I like what you're looking at here. At the end of cleavage, this solid sort of mass of cells, they give it a name. We looked at it in lab. This was called the morula. Again, it's just named because what it looks like. It looks like a mulberry with raspberry, blackberry, all those little compound mulberry fruits. That's what the embryo looks like, according to the people who first described it. They named that the mulberry tree. That's the morula. That's the end of cleavage. I'll put that in as a stage, sort of. So cleavage stops there. Now mitosis, of course, doesn't stop. It's going to continue till you're dead. But what's going to happen next is you will begin to see some growth. So the next phase, I'll show you over here on the right. This right here would represent the morula. It's just a solid mass of cells. The cells, some of them are connected with tight belt desmosomes, so they won't separate. Others are free. What happens after you get this morula, the embryo starts to expand in size. It basically secretes fluid, and it swells up like a balloon, and you get this thing right here, which if I were to draw, it looks something like this. This structure is called blastocyst. You could describe it as a hollow ball of cells. This space here, what they've labeled a plastocele, it's just an opening, that's fluid. So this is like a hollow ball, and it kind of looks like a follicle, like looking at a thyroid or ovary. It's this big hollow ball, fluid in the middle. Notice though they shaded the cells with 2 different colors. In fact, in a lot of these diagrams, you will see a particular color pattern. Don't get too worried about it. We don't do much with them, but let me stick this up here real quick. Early development, the cells are all the same. They have not specialized at all. If you look at them on the microscope, they look the same. They haven't expressed any differences. What they do, sort of classically with these diagrams of embryos, they use some things called fate maps, and that's where they get these colors from. What I mean by that is, all of these cells, if you look at them, they look the same. But notice they've shaded some blue, some red, some yellow. The reason they can do that is they take these cells in an embryo, let's say these cells over here, they mark them. They used to use India ink. They inject it sort of into the individual cells. India ink is a particulate ink, simple procedure. But then they can follow that cell and see what it develops into later. So they know sort of in advance that these cells here, if you watch them, they're going to develop certain structures. These ones in blue, they'll develop different structures. And same with yellow. So that coloring, while it's nothing you can tell looking at the embryo, the embryologists have determined what those cells are going to do. They have a particular function later in the adult, and we know that because of past experiments, and it's pretty regular. So that's what the colors mean. They aren't, there's really nothing different that you can detect with a microscope. So that's why you got these 2 different colors of cells. These are 2 different populations of cells, and if you follow them, they're going to produce different structures. In fact, these two populations of cells, if you look at in the lab, this group here, this kind of massive cells sort of inside the structure, that's usually called that is the embryoblast. So these kind of orange cells on this diagram. The blue cells, those are referred to as the trophoblast cells. They are all the embryo cells. If you took them out, look at the nucleus, look at the DNA, they're not the mother cells, they are the embryo cells. They are fetal cells. However, what they develop into is going to be different. So the reason they made this subdivision in these diagrams, this clump of orange cells in the middle, the embryoblast, that's going to make up the embryo. That'll be its arms, its legs, its guts, its brain, all its parts. This outer sort of blue shaded cells, what they call trophoblast, even though they're embryonic cells, they're not going to make up any part of the embryo. Kind of look at this in lab. These are going to be the extra embryonic membranes. They're support structures, things like that amnion and the chorion, allantosis and such. They're going to be support cells. They never are part of the embryo. End up being afterwards. That is the blastocyst. And one other thing that will begin here, if we look at this embryoblast now, we'll ignore the rest of it. It's kind of looks like a little sort of sandwich of a couple layers of cells. These cells have some interesting properties at this point. I should be using different colors. Let me change that. You'll notice even in the models in lab, if you look at the late stage blastocyst, they actually use two different colors to describe the cells of the embryoblast. So I'm not throwing in the trophoblast, that's just kind of that bit there. When you look at the embryoblast cells of an early trophoblast or early blastocyst, these cells have some unique properties. They are these three terms mean the same thing. Totipotent, indeterminate, or sort of more generally, embryonic stem cells. Typically in most general magazines, newspapers, they'll use the term embryonic stem cells, so they sort of all mean the same thing. These have unique properties. In fact, there's controversy around these embryonic stem cells. There are some people who want to do research and things with it. Other people say, hey, that's an embryo, don't do it. But what do they want to use these embryonic stem cells for, Any particular use for these things? Why do people want to do research with them? Cell research, yeah. Why these?
Isn't it like cell division they can do?
What they can do, this term indeterminate, what it kind of means is the developmental fate of these cells has not been determined. Indeterminate. What it means is if I pull a cell out of this embryo or separate it into two piles, one cell is able to develop into the entire embryo. That is, if you take this mass of cells and you can split it in half, like they did this early experiment with these embryos, generally not human, again, they're hard to work with. They take this embryoblast, they tie a little hair around it and a knot and go split it in two. You get two embryos developed completely intact. This is actually how you get, you look at twins, there's fraternal twins, identical twins. Fraternal twins are not a big deal. Yes, they're just two eggs fertilized by two sperm. They're just siblings. That's why you can have a male and a female. They share about half their genes, like all siblings. They just have to be born at the same time. Identical twins are different. They're genetically the same. This is what happens if somehow this embryoblast is split in two parts. You'll get two embryos formed, but they start from the same sperm, same eggs. They're genetically the same. So that's the term identical twins. So these cells they still have access to their entire genome. So in theory, you can do things with them. The ideas are, hey, you got bad heart cells, we can take these stem cells, convince them to develop into new cardiac muscle cells. There you go, replace the damage. You got some bad neurons, here we go, we'll put some stem cells in, boom, neurons. That's the idea. They have a little success with it, but they haven't been able to do all that yet. But that's kind of why they want to use them. They have the ability to develop into anything. This is also the stage, the blastocyst stage, because this is still in the uter tube. You know, if you look at fertility treatments, in vitro fertilization, things like that, you can get an egg, you can fertilize it, you can get it to develop to this stage because you don't need a uterus yet. Those are the ones they freeze in fertility treatments. You know, in fertility treatments, they take eggs, they harvest them from the female, they fertilize them, then they implant them back into the uterus at this stage. Of course, they don't implant one or two because the success rate isn't that high. So we'll put in a bunch, hoping that one or two will implant. Occasionally all 8 implant or something, and that's when you have those octuplets and things like that. But extra ones they have for fertility treatments, they freeze them. These are small enough, you can freeze them liquid nitrogen, they'll keep forever. But that's kind of how they're stored. That's the part you get from in vitro fertilization, because you don't need the uterus yet. Now in humans, the early blastocysts, those cells are what we call embryonic stem cells, where indeterminate. If you wait to the late blastocyst stage, that ability is gone. What happens, even this early in development, these cells lose the ability to develop into anything, and basically their developmental fate starts to become locked in. This is starting down the pathway of what we call specialization. You know, last semester you looked at specialized tissues, right? If I gave you a skeletal muscle cell and I gave you an epithelial cell on slides, could you tell the difference? Assuming you go back and maybe check your notes from last semester. They don't look anything alike, right? Big cylindrical skeletal muscle cell, little flat squamous cell. But here's my question. Genetically, what's the difference between your skeletal muscle cells and your epithelial cells? Do they have the same genes? Yeah. So there's got to, too. So how come they look so different? They're identical genetically. Should they look the same? Yeah, they don't. But why don't they? Because what happens as you go down the path of specialization to make epithelial or skeletal muscle cells, the cells lose access to some of the genome. For example, if you're a skeletal muscle cell, do you need access to the genes that make teeth, hair, and nails? No, because that would be really uncomfortable if you were expressing crunch, it wouldn't work. So that's what happens as the cells specialize early on in that sort of pathway. The tissues, the cells, lose access to genes they don't need. And so if you take an epithelial cell, the only thing it's able to grow is basically epithelial products and other epithelial cells. You can't make an epithelial cell develop into a skeletal muscle cell. That's why they want the embryonic stem cell. They can produce anything. So what happens in the late blastocyst, the cells are starting down that path to be specialized, cells. Now they're not there yet, but their cells will begin to lose access to some of the genome. Now if you looked at them, they weren't expressing the differences. They still all look the same at this point, even though they might shade in different colors. That's because we know what they're going to do. But they haven't expressed differences yet, but there are going to be some changes in what access they have to genes. So the first thing that happens in this sort of pathway to specialized cells, and it happens by the late blastocyst, is we start to produce some things called embryonic germ layers. So by the time you get to the late blastocyst, these cells are not, sort of in quotes, the embryonic stem cells. They're not totipotent cells. They're going to start to lose access to some of the genes. And the first two sort of populations of cells that will emerge and become some of these embryonic germ layers will be these embryonic germ layers. There's only 3. So these are populations of cells that will develop in the early embryo, beginning in the late blastocyst stage. Sort of the first 2 that more or less form. One's called the ectoderm, one's called the endoderm. To be completely correct, I have to say it's presumptive ectoderm and endoderm. They're not quite there yet. They're close enough. I'm going to call it. All right. Typically, ectoderm on these models I'll be showing you, those cells are shaded in blue. So when you look at that frog embryo back up. There's a frog, not a human, but more or less the same. So when you see these blue cells, that's actually representing ectoderm. These yellow ones are representing a different population, a different embryonic germ layer. That's going to be the endoderm. So in the blastocyst, what you end up with It's kind of one layer of cells, we're gonna call it the ectoderm. It's presumptive ectoderm, but it's pretty much there. The other layer of cells, I don't have yellow, so I'm using black. That would be endoderm. Now, what do we need to know about these embryonic germ layers? Again, their developmental fate is fixed. So at this point in human development, if you took a late blastocyst cell and you pulled out some of these blue cells on this top, one called the ectoderm cells, and you had the ability to grow them in some kind of tissue culture, you would not grow a whole embryo. The ectoderm would only produce the integument, the skin, and all associated skin structures. So it can grow your skin, your hair, fingernails, teeth, horns, yeah, that sort of thing. So ectoderm will produce the skin, the integument, and the nervous system. So the ectodermal cells, if you grew them, you basically get a bag of skin with brains and spinal cords in it. That's all you get. You wouldn't get any other parts of the embryo, so its developmental fate has been limited. If you pulled out some endodermal cells, and did the same thing, again, you wouldn't get a whole embryo. What you would get with the endoderm is you would get the digestive system, you know, the alimentary canal from your mouth to your anus, that big 30-foot tube, and the digestive organs, liver, pancreas, gallbladder, and so forth. And it would also develop into respiratory structures, which really just develop as an outgrowth of the digestive system, so your airways and your lungs. So if you grew endodermal cells, that's it. You get kind of part of your guts, nothing else. Now there's one other embryonic germ layer. It won't develop until sort of the next step, but I'm gonna go ahead and list it now. I got the other two up on the board. The third embryonic germ layer, which will actually form in the next developmental stage, is called the mesoderm. It'll produce everything else. Basically, you got your guts and you got your skin. You don't have anything else. So mesoderm is going to produce your skeletal system. I'll just put down bone, it's easier to write. Your skeletal muscle, your blood, and blood vessels, heart, kidney, all your organs except the digestive organs. So it's basically all the stuff in between. Your endoderm makes your gut, your alimentary canal, your digestive system. Ectoderm makes your skin. Mesoderm kind of makes everything in the middle. So your skeletal system, your muscle, your blood, your heart, your kidneys, and so forth. That'll be mesoderm. It hasn't quite developed yet, but that will be the third one. So those are the three embryonic germ layers. Two of them start to separate out by the time you're at the late blastocyst. So let's move to the next phase. All right, the next process, let's go along with the next one. Right now, what we've got is an embryo that looks like a flattened disc. I mean, it's basically two layers of cells. If you look at it in cross-section, it looks like this. If you looked at it now from the top, it would just kind of look like an oval. It's actually sometimes called an embryonic disc. It's just kind of two layers of cells sort of sandwiched together. Let's see if I got a picture here. Sort of like this. So we're looking at like a 2.5D representation. Here's that top layer, here's that bottom layer. The next stage in development basically has to take this embryo, and it does a couple of things. It allows the third embryonic germ layer to form at mesoderm, which isn't there quite yet. And it also gives the embryo its adult axis. You know, your adult design is not like a flattened disc or a sandwich or something. You know, you're a tube-like thing. You got a head end, you got a tail end. This thing doesn't. So the next stage is going to form the third embryonic germ layer, and it's also going to give the embryo its adult body plan with a head end, a tail end, a left and right side, and so forth. This next stage is called sorry about some of these names, I didn't invent them. Gastrulation. The structure is called a gastrula. Actually, what you're looking at right here. All right, what's happening here? Draw a picture, and we'll use this diagram. So, We're starting off, this is a little later than the blastocyst, but you basically have a layer of cells here and here, kind of what I got here. So imagine this is a cross section. Make it a little longer. And if you're looking at it from the top, this direction, it just looks like a disc. This one's kind of tilted at an angle. Here's that top layer, bottom layer, yellow. That's all that's happening. Well, what happens in gastrulation, if you watch the cells, these cells are not locked in place. They can move. So they will literally start moving, and the direction they're going is right towards the midline. So these cells are going to move this direction. These cells start moving this direction. That's kind of what they're trying to show with the arrows. All these cells on the surface are moving towards the middle. You can actually see this on microscope slides of the embryo. What you'll see is a couple of dark lines appearing in the middle of the embryo. Why do you get dark lines there? Because as these cells migrate towards the middle, they basically sort of start to stack up here, which makes a couple of darker areas on the microscope slide. So you can see these two things are actually kind of ridges of cells here. They call it primitive streak. You can see it run down the embryo. What's happening, those cells, as they migrate towards the midline here, they basically push down and in. Again, these cells are mobile. They're not locked in place at this point. So you can imagine as these cells pile up, some of them are going to roll into the inside, and they move into this space between the upper layer here, lower layer here. These cells are literally rolling into this sort of groove, and they break off. And now you'll notice on this diagram, they change them to a different color. This is the mesoderm. This is how it forms. So the cells that are going to be ectodermal cells, they start migrating towards the midline, they break off, and they actually fill in this space. So now what you've done, you took a two-layered embryo, and it's now a three-layered embryo. It's got a layer on the top, layer on the bottom, and now these mesodermal cells have filled in between. These are the three germ layers. Again, looking at them, you couldn't tell the difference. They haven't expressed the differences yet. They're not specialized cells yet, but they are losing access to the genome, so we know what their developmental fate is going to be. It's been kind of locked in already at this point. Now we get to this point. Back up here, look at this one. So here's where we are on this slide. Sorry, I'll pause it, this magnification. Here's that blastocyst. The uterine tube is up here. The blastocyst is what makes its way into the main body of the uterus. The blastocyst is what's going to implant, and that's what they show you here. As the blastocyst comes in, notice, see this little white membrane that's peeling off here? At this point, this is when as that blastocyst expands, it actually sheds that fertilization membrane. We're referring to it as hatching. Of course, it's not really hatching, but as that blastocyst expands, that fertilization membrane, which isn't very stretchy, it just tears and pops off. Now, once that fertilization membrane is gone, now the blastocyst is free to implant, and this is the endometrium here, so the blastocyst is embedding into the walls there. The embryo we've been looking at is right here. So that's the embryoblast there, and it's basically an implantation. It's like those two layers of cells, the ectoderm and endoderm. Shortly after implantation, that's when the embryo undergoes gastrulation, right here. So this is shortly after implantation, the embryo is implanted, those two layers have formed into three. We're not done with gastrulation though, because what we've got at this point is a three-layered sandwich. That is not the adult architecture. Here we go. So here you are. This is kind of what you look like at this point. You're a little flattened disc, ectoderm in blue, endoderm in yellow, mesoderm, the red in between. So it looks a bit like a sandwich. The last thing that happens in gastrulation, well, that's just visualizing, but you're kind of like this. So imagine this is your endoderm, this is your mesoderm, this is your ectoderm. So that's what you look like right now. You're just a little sandwich, three layers. What happens now, we're trying to show at this bottom, the whole embryo just kind of rolls like this into a tube. And when it does, that's the adult architecture. If you think about it, this tube in the middle, right here, the endoderm, That's your intestine. Mouth at one end, anus at the other. Then you got this thin layer, the mesoderm. That's all your muscles and your skeletal system. Then the outer layer's gonna be your skin. So, and that's what they're showing you right here. So once this thing rolls up, you've got this hollow tube, that's gonna be your digestive tract. Mouth at one end, anus at the other. The ventral surface is kind of where you seal. If you were flat like this, you would roll up into a tube. So you'll have this kind of suture running down the middle. That's in your front, that's your belly, dorsal surface. So now you have an embryo. It's got the 3 germ layers, and it's got a head and a tail head, the left and right side, a dorsal ventral surface. It's kind of the basic architecture. Oh, put it up. That's the end of gastrulation. Now, at about the same time, there's some other things going on. Remember, we've just been looking at the embryoblast. What about that trophoblast, those other cells? Let's look at that next. Now, the next structures that we look at, they kind of form at different times. I'm just lumping them into a pile. Don't think they all form at the same time, they don't. But as gastrulation occurs, you see one little envelope forming out here. That's going to be the amnion. But let me switch back to a better picture. It's a little later in development, but what I want to look at next is what the non-embryo parts of the embryo developed into. These extraembryonic membranes we looked at in lab. Let's talk about those in a minute. And let me mention a couple things about them. I need to put it in context, so I talk about chickens a lot when I look at these extraembryonic membranes. Kind of like I do the same thing in lab. And there's a reason for it. Well, you don't really need to know anything about a chicken. If I just talk about humans, these membranes don't make any sense because they don't do anything, or some of them don't do anything. So it's different. All I'm going to say is, if you look at vertebrates, animals with a backbone, that includes fish, frogs, salamanders, reptiles, birds, mammals. Those are basically the vertebrates. That is an evolutionary lineage. They split the vertebrates into 2 categories. There's one group called non-amniotes, and there's another group called the amniote lineage. The non-amniotes are your fish, your amphibians, frogs, salamanders, newts, and so forth. The other lineage, the amniote lineage, are birds, reptiles, and mammals. The difference in those 2 lineages is the type of egg they produce. The amniote lineage produces something called an amniote egg. You're familiar with it. It's a chicken egg. The non-amniote lineage does not produce an amniote egg. You may be familiar with those as well, like frog eggs or salamander eggs you might find in the pond. Almost about this time of year, they'll start going out to the ponds and laying these jelly-like masses of eggs. Those are non-amniotes. The main difference evolutionarily is if you take a frog egg, or you take a fish egg, or you take a salamander egg, and you put it on the sidewalk on a nice sunny day like today, what happens to it? It dries up into a little shiny crust in a couple hours. They dehydrate. Those non-amniote eggs, like fish eggs, frog eggs, are basically the same. They have to be put in an aquatic environment, or at least a non-desiccating environment. If it's dry outside, they lose water immediately and the embryo is dead. Not very good for your reproductive success. That's a problem if you're trying to exploit a terrestrial environment. So what you have in the amniotes, they produce this amniote egg, and the main difference is the amniote egg can be left in a desiccating environment for months, and it doesn't dry out. Think of those like Komodo dragons, the biggest lizard we've got on Earth right now. That has an incubation period for its eggs about the same as our gestation period, 9 months. So that female plunks its egg somewhere, and it just sits there on the ground for 9 months, and it doesn't dry out. It's not in the water. In fact, they don't even develop in the water anymore. But what they have in these amniote eggs are a series of extraembryonic membranes, and their main purpose is to allow the embryo to be in a dry environment, and the embryo will still develop and hatch. Now, mammals are part of this amniote lineage. In fact, mammal eggs, there's too many egg-laying mammals left, but there's a few. The ductile platypus and the echidna are egg-laying mammals. If you look at their eggs, it's just like a chicken egg, just like a lizard egg. They're all the same. Now, most of the more modern mammals, including us, we are placental mammals now. We don't lay eggs anymore. However, we retain all the same membranes that an egg-laying lizard or a chicken produces. There's not that much different. Ours have just picked up some additional functions, which we'll talk about a little bit less. So I like to make the comparison between a chicken and a human. And in fact, if you look at these early embryos, you really can't tell the difference between a chicken and a human embryo. They pretty much look the same. We develop almost exactly as a chicken. There's not that much difference until you get to later developmental stages. Same with lizards, develop the same way. Early embryos, They all look the same. Almost the same. So I'm going to use the chicken because it's kind of got the basic function for all membrane still. It's an egg layer. And then we'll just kind of contrast that with a placental mammal or human. And again, we went over these in lab, so it's sort of the same thing. But the outermost layer on this diagram, this thing called the chorion, kind of shaded in pink, its original function was basically gas exchange. I mean, think about it. You got an egg, and you got this little embryo in there. Embryos have metabolism just like you, and they use cell respiration to make their ATP. So they've got to have good supply of oxygen for the electron transport chain down to the mitochondria to function. The problem is, if you look at some of these eggs, like look at an ostrich egg, the oxygen is way out here, outside of the egg, right? The distance between the atmosphere and that embryo inside that egg, that could be huge. It could be like 10 centimeters. Is oxygen going to diffuse that distance? Goodness no. Again, diffusion is really slow after a few cell diameters. It can take years to get in there, so that won't work. So the cell doesn't have, or the egg doesn't have the surface area, and the distances are too great, you can't supply the embryo with oxygen. So what do? Well, as this embryo develops, gets bigger and bigger, has a more demanding metabolism, it will grow some blood vessels. We do the same thing. We have umbilical arteries and veins. The bird, they call vital arteries and veins. Same thing though. The bird's circulatory system will pump blood out through a membrane that you'll find just under the eggshell. It's called the chorion. And as that blood passes through, the shell itself is very permeable. That's got holes all over it, so that doesn't do much anything. But the oxygen diffuses into the blood vessels in the chorion. It picks up oxygen, and then it makes its way back to the chick embryo. You can see this if you have, if you've ever hatched chickens or raised chickens. How do you know an egg's fertile? After a few days, if you take that egg and hold it up to a bright light, they call it candling the egg, so you can see if it's any good. If you look you'll see just a dense mass of blood vessels in there. That's the chorion. Right under the egg shell, there's going to be this dense mass of blood vessels. They grow in the chorion, and that's what the chick pumps blood through. So as it pumps blood through that chorion, CO2 can diffuse out in the atmosphere, oxygen diffuses in, the oxygenated blood then returns back to the embryo for gas exchange. Now the difference between the chicken and a human, or any of the placental mammals, we still have a chorion. And we still do the same thing with it. The embryo will pump blood through umbilical arteries to the chorion, and then it will come back in veins to the baby's circulation. But the difference is, what's outside of the chorion in an egg layer? It's the atmosphere, right? Oxygen, nitrogen, that sort of thing. What's outside of the chorion in a placental mammal? It's not atmosphere anymore. What's out here? This is maternal blood. All of this is the endometrium. And blood sinuses that are filled with maternal blood. So this part right here, that's the endometrium. And of course, as that embryo implants, that trophoblast that grows into the endometrium, it actually damages it. You got a lot of damaged tissue and broken blood vessels. So you end up producing a lot of blood sinuses. So that white area is actually where maternal blood would be pooling. So that endometrium and that chorion with its villi, that is the placenta. So what's outside of the placental mammal's chorion is maternal blood, not atmosphere. Now, what does that mean? Well, does maternal blood have oxygen in it? Yes. So oxygen will be free to diffuse from the mother's blood into the fetus's blood, same way as a chicken. CO2, same thing, just a different direction. So the chorion is still used for gas exchange, whether you're a chicken or a human. But here's where it differs a little bit. The chicken, or any of the egg layers, can't do anything else. There's not much in the atmosphere except some oxygen you can use, and you can dump CO2 there. Well, what's in the maternal blood? Yeah, it's got oxygen, great, we use that. We get rid of CO2. Anything else in blood that's useful? Yeah. Mother's blood is full of things like nutrients. There's some glucose molecules. You got amino acids. All of these nutrients can also diffuse across the placenta fairly easily. So it's picking up additional functions. In a chicken, it's basically just gas exchange. In a placental mammal, it is gas exchange, but it's also acquisition of nutrients, and then other waste material like ammonia, nitrogen waste, ammonia, urea, things like that. They can be dumped across the placenta, given to your mother's blood to take care of. So the chorion has picked up additional functions in the placental mammals that aren't present in chicken. They're both used for gas exchange, but the chorion in the placental mammals has picked up additional functions. All right, that is the chorion. Now the next one. Growing out of the ventral surface, there's a little membrane, little bag-like structure that will encase the entire embryo. Whether you're a chicken or a human, you'll have this bag around you, and it is filled with fluid. It's amniotic fluid. What's its function? Well, remember, those vertebrates used to lay their eggs in water. Water's a great place. It's a great medium to lay your eggs in. It's fairly stable. It's also very dense. The problem with having no water now, because these eggs are laid in the terrestrial environment, is you don't have the buoyancy of the water. And the problem there is like, can anyone in this room raise yourselves up on one finger? No. Could you do it in a swimming pool? Yeah, it's easy. You weigh almost nothing in water. The density of your tissue is almost the same as in the water. So it's almost nothing. You're almost weightless. That's partly what the amnion is for, the amniotic fluid. Because the eggs aren't laid in water anymore, this membrane is basically taking the water with you. So even though you're in a terrestrial environment, these embryos are developing in basically that weightless environment in the water. So that buoyancy of the water in the amniotic fluid is important. Otherwise, if you didn't have it and you developed on the surface, gravity can push you flat, but it'll be flat on one side. It'd be maybe easy for getting into elevators and stuff, but it'd be a little inconvenient. So you basically develop in this weightless environment. It does the same thing in a chicken and a human. You develop in this fluid environment, which the density makes you develop pretty much in a weightless environment. You don't have to worry about gravity squishing you flat somewhere. It has some other minor roles. A layer of fluid is good at absorbing impacts. So, if the eggs kicked around or mom falls down the stairs, that water tends to dissipate impacts over a wider area, so it'll do less damage. So it does have some other protective roles. It may be some thermal protection for the chicken as well. That's not an issue for placental mammals because mom's thermoregulating. But it's mostly so you develop in a weightless environment. So both chickens, placental mammals, humans, that's about the same. And then let's do the yolk sac. Always shaded yellow in models, which is a dead giveaway. Of course, in the chicken, you got an embryo, it's going to be developing for 3 weeks of your chicken, 9 months of your Komodo dragon. Those embryos need an energy source and they need raw materials. So, of course, the yolk sac is just a membrane that forms around the yolk. Yolk is just the supply of raw materials, nutrients that the embryo needs for early development until hatching. That's what the yolk sac is for. It's just holding the yolk. In placental mammals, of course, that function isn't necessary because the egg layers, you need the nutrients. There's nowhere else where we come in. But because the chorion has taken over the role of the yolk sac, human eggs don't have any yolk. The yolk sac is somewhat vestigial, not completely, but you don't have any yolk anymore because you don't need it. You get all your nutrients and raw materials right across the chorion, across the placenta, so there's no need for yolk. So while the yolk sac is still there in the human embryo, there isn't any yolk in it. The only function I can give you for a yolk sac in humans is it's a site of early germ cell production for, blood cells start off in the yolk sac. Cells in the yolk sac produce your first blood cells that then migrate to the embryo proper, and they end up taking up residence in the bone marrow. Some of the reproductive germ lines are in the yolk sac. I think the precursors to your sperm or eggs, that population of cells, I believe, begins in the yolk sac and just migrates to where the testes are going to be a little later in development. So there's still a few functions for the yolk sac, but it's really not yolk storage anymore. It's lost that function because the chorions basically picked it up. And that goes for the last one as well. Kind of shaded in green here, pretty big in the chicken. Not much in a human embryo. This is the allantois. What its original function is for, think of a chicken or a Komodo dragon sitting in there. Now, what's going to happen is you're going to be metabolizing amino acids. When you fully metabolize proteins, you can burn them, they're an energy source. You can only use that bit. You can run it through your metabolic pathways. What you're left with as a waste product when you metabolize proteins is this stuff, NH3, that's ammonia. You can call it generally nitrogen waste. So all animals are going to produce some nitrogen waste, particularly when you're metabolizing proteins. The problem with nitrogen waste is it's really toxic. Ammonia will kill you. So most of these amniotes and us, we convert the ammonia to another nitrogen waste form that's less toxic. We produce urea. Your liver converts ammonia to urea. And most of the other amniotes produce something called uric acid. But it is still toxic. You got to get rid of it because it'll damage the embryo. So what do? With all your nitrogen waste, urea for us, for 9 months if you're a Komodo dragon? It's going to build up in here and poison the embryo. So this allantois, it's basically a waste storage sack. Because we can't open up the egg and throw your junk out on the neighbor's yard or something. It's all sealed in. So the allantois is just a package, an area to store the nitrogen waste until hatching. It keeps it away from the embryo. That's mainly its function. In humans, the allantois is nearly non-functional. Why? Because what do we do with our nitrogen waste? We don't need to store it anymore. We dump it across the placenta, let mom's kidneys deal with it. We don't have to store it. So the allantois has lost most of its function. In fact, it doesn't really do anything in a human anymore. I think the only function I can find for it is part of your urinary bladder forms from the allantois, but it really doesn't do much anymore. It's fairly vestigial. That's in humans. Some other placental mammals, like I think a horse, the allantois is actually really big, and it fuses with the chorion, and it actually forms part of the placenta. So you can have a chorioallantoic in some placental mammals, but it's a secondary function, not waste storage anymore in the placenta. And ours is very reduced. There's only a remnant of it in the umbilical cord. So as far as the extraembryonic membranes, know what they do in a human. I don't care if you know what they do in a chicken, but I don't know, I kind of like to do that because otherwise I don't even talk about that allantois. It doesn't do anything in humans anymore. But that's why it had a role. It was somewhat vestigial. It lost some of the functions of the chorion because of the formation of the placenta. Now, last bit. All right. I'm going to go over one more thing. We'll call it quits on this. Let's look at. So about this embryo now, he's rolled up into a tube after gastrulation. Of course, the chorion was there already. The chorion formed from the trophoblast. It just grows and develops a bit. We start calling it the chorion. Amnion, yolk sac, and allantois, they develop shortly after gastrulation. Those embryos will be present. They'll just grow in size. So what we've got now is an embryo after gastrulation that basically looks like a tube. If we look at cross section, it might look something like this. There you are at this point. So after gastrulation, you roll into the tube. So the outside of the tube is ectoderm. The inner wall, that's endoderm, and then between it, you got a layer of mesoderm. So you just kind of a coiled tube with a hole going through it, your mouth, your anus. None of the cells look different yet. Again, on the diagrams, they'll all be color-coded and such because we know that they're gonna look different, but they haven't expressed differences yet. So there's really no structures that you can identify in this early embryo right after gastrulation. That's about to change. Shortly after gastrulation, cells will start to express some different genes, and they will start to form structures you can identify. So let's go over a couple of those here. I'm going to stick a frog embryo because I don't have to do human, but it's the same process. What will happen early in development, some of the cells in the mesoderm, this one's called pretty much right here. So the red is the mesoderm, yellow is endoderm, and this kind of blue is ectoderm. Some of the cells in the mesoderm will start to condense and form a cartilaginous rod that runs from the head to the tail of the embryo. If you're looking at a sort of longitudinal section, there you are, you would see this rod begin to form out of that sort of amorphous mesoderm at this point. This cartilaginous rod kind of runs from the head to tail. It's the main axial support of the embryo. It's called a notochord. It sort of takes the place of the vertebral column in the embryo. It's an axial support. It's a flexible rod, but it won't compress much. So it gives it some sort of axial support. Later development, of course, the notochord gets replaced by the vertebrae, but there are remnants of it still there. You know those intervertebral discs that are between the vertebrae? There's a little fluid-filled, pouch in the middle of that. That's the remnant of the notochord. The vertebrae just grow through it, basically displace it. There are only remnants in adult. But early in development, if you look at a human embryo, it's a notochord before it's a vertebral column, but they're in the same spot. Now, what's interesting when that notochord forms, it's going to induce the formation of really the first organ system. One of the first organ systems that begins developing in embryo is the nervous system. It's also the last to mature, so our nervous system takes a long time. Very early development, first thing you'll notice is nervous system beginning to form, and then it takes, 21 years before it's finished? So some of you might still be working on it. But what we're looking at, do my ectoderm here, and here's a little notochord. There, here's endoderm here. All right. This process they're showing, this is basically going to be the first organ system that forms. It is the nervous system. The process is called nervulation. It's the formation of the nervous system. Remember, doesn't it seem weird? The ectoderm is the skin and the nervous system. That always struck me as weird. There aren't a lot of organisms that develop that way. But how does the ectoderm, which is on the outside, produce the nervous system, which is on the inside? This is how. It's a very unique developmental pattern, only found in the group of organisms called chordates. That's all the vertebrates and a few others. What you'd notice if you observe this embryo developing, you'd see that little notochord form, and then you'd notice the ectoderm right above it begins to thicken. The cells proliferate a bit, and you end up producing a thick area just above the notochord. This thing is called a neural plate. Kind of what you see right here. Now they've changed the color from blue to green here, but this is a thickened layer of ectoderm. They call it the neural plate. So it's got this thick sort of flat mass growing above the notochord in the ectoderm. Then if you keep watching it, what's going to happen is that thick plate starts to sink in the middle. What you're seeing here, again, use that notochord as a landmark. Notice that neural plate now has sort of sunk in. It's made a U or a V-shaped structure here. That, where it's dipped down, you can actually see a groove along the back of the embryo. This is usually called a neural groove. And you'll see a hump on either side, here and here. They call those neural folds. And these are visible externally. If you look down at these embryos under the microscope, see these two kind of humps form with a groove down the middle. That's it. Those humps, the cells are kind of sinking in. Eventually they'll sink in completely and seal off. So the next thing you might see is something like this. So that whole sort of plate just sinks in, and eventually it sinks completely below the ectoderm, and it will eventually sort of seal over. There's your little notochord. And this is now called the neural tube. This is your brain and spinal cord. It's kind of a weird way it develops. If you think about it, your brain and spinal cord start off as a flat patch of cells back here. It's just sank in, rolled into a tube, pop, and kind of pinched off from the rest of the ectoderm. That's why there's remnants of this. If you remember looking at the brain last semester, you looked at the ventricles, like the lateral ventricles and the third and fourth ventricle and the cerebral aqueduct. All those hollow spaces filled with cerebrospinal fluid. That's because in here, your brain and spinal cord start off completely hollow. It's because they start as a tube. I mean, they rolled up into a tube. That hollow space, it does fill in with cells, but there's remnants of it. The ventricles of the brain and that central canal of the spinal cord, that's because of the way you develop. Your spinal cord and brain start as a flat sheet that rolled into a tube. You start off as a hollow tube, and then, of course, it'll slowly, almost completely fill in with cells. This is also where you get like for spina bifida, that's a neural tube defect. Sometimes there's something wrong and that neural tube doesn't finish sealing closed, so it stays open, that's what spina bifida is. It's failure of that neural tube to roll up completely and end up where it's supposed to be. What's kind of cool with stuff like this, we won't do much more, but this is really predictable. I mean, the timing of these events, the order they happen is the same every time. And so what we know about development, of course, we're learning more all the time. The reason development is so ordered, why do certain parts develop in certain areas? Just kind of in a nutshell, when you look at that first egg, remember, the egg supplies everything to the embryo except 23 of the chromosomes of the human, right? Well, the cytoplasm in that egg is not uniform. You imagine here's a big egg cell to start with. There are concentration gradients of various chemicals. I'll put some there, and then I put some red ones here. And then there's some black ones. It's not a homogeneous mixture in the egg cytoplasm. And then when that cell divides by mitosis, the cells that end up forming don't have the same components in their cytoplasm. So because all the cells are a little different chemically, it's those chemical concentrations that sort of signal a particular cell to produce a certain structure. And then there's a lot of signaling between groups of cells. Like, for example, development of the nervous system, brain and spinal cord, they did early manipulative experiments. If you take an early embryo at this stage, you cut out the notochord out of one embryo, and let's say you implant it in this embryo. So this embryo has a notochord here, a notochord here. You know what happens? This embryo will develop with two nervous systems. You get a brain and spinal cord developing there, and one up here. What that tells us is there's some kind of signal. When that notochord forms, it is somehow sending some kind of signal. And we know what some of these signals are. They can be things as simple as calcium concentrations, or there's CCK and a bunch of other chemicals that can be released when a certain structure forms. They act as a signal, and that's what signals the overlying ectoderm to develop into the brain and spinal cord. If you transplant another notochord in here, it'll just signal another set of ectoderm to develop into a spinal cord. There's all kinds of signals and concentration gradients that direct development, so it's very ordered and very predictable. It's like, what's one of those signaling substances? It's got a cool name. Sonic Hedgehog. You know Sonic Hedgehog, the video game guy? Well, not too many years ago, they found a group of cells in the appendages, basically, that have a gene that produces a particular protein. They call the gene Sonic Hedgehog. I don't know why exactly, you can insist on telling. But that's what makes your fingers form. So it starts off at one area. There's a patch of cells that release this protein. And as it diffuses away from the site, the concentration gets lower and lower and lower. So if the concentration is really low, you get a pinky. If it's a little higher, you get this one. A little higher, you get a middle finger, and so on. That chemical, if there's too much of it, that's why you get people that are polydactyl. They got extra fingers because it's that one signaling substance. If it's at a higher concentration, you may develop extra fingers. They can trick embryos into producing extra sets of arms. And it's all these chemical signals. That's kind of where a lot of the work moving in embryology is now. It's how these chemical signals work and what they are and kind of how they're regulated. But that's just kind of one example. So you got that notochord, it's signaling the overlying ectoderm. Wherever that notochord is, you're going to get a nervous system to form. That's good enough. That's all. Yeah, we got a nervous system. Everything else just going to grow. Actually, the heart grows next. In the early embryo, you'll see the heart form. It starts off as a tube, then it rolls into our more recognizable, heart with blood vessels going in one end. But then after about 8 weeks, you got all your organs. That's the end of the embryo. After that's the fetus, and then it gets more. It just grows, gets bigger, grows, gets bigger, grows, gets bigger. So I'm not doing any more. So I'm done with all I'm going to do to you with development. So what are we going to do next? Let's go over the exam. All right, format for the exam. Of course, I don't know what it is, I still have 2 days. But it will be, I don't procrastinate, never. Probably somewhere in the neighborhood of 50 or somewhere around 50 multiple choice fill in the blank type questions. And they're typically worth 2 points each. So that's 100 points there. There'll be, let's see, we have 1 quiz, 2 quizzes, right? We have 1 or 2 quizzes. Can't keep track. I got an 8-week section going at 60, just the one. And that was mostly fill in the blank I gave you? Yeah? Okay. So you will get, multiple choice fill in the blanks, similar to the ones you have in Quizlet, a little both. Short answers, you will get a handful. Probably if I gave you, let's say, 6 short answers, 3 or 4 would be the endocrine, and then the last 2 or 3 would be everything else. Let me give you some examples. Let's see, we're endocrine system. I can tell you what short answers will be. Or I won't tell you what they'll be. These are short answers I've used in the past a lot, which means they're likely to pop up there. Number 1, just like on the quiz, I will give you a gland. Probably not a posterior pituitary since I already used it. And I'll just say, list all the major hormones secreted by the blank gland. For each hormone, list its action and list the target. I could do something like that. Or I can just give you a list of hormones, random hormones, and say for each hormone, list its origin, its action, and its target. Oh, yeah, stuff you gotta know anyway for your multiple choice questions and everything else. So, gland. Hormones it secretes, plus their action, maybe their target, that sort of thing. That's 1. Number 2. Oh gosh, there's so many options for the endocrine system. I could ask you something like, something along the lines of, all right, explain how the cyclic AMP second messenger system works or functions or something like that. And you just do it in steps. When you answer my short answers, you can do in text, you can make tables, whatever kind of works for you. I just need to know you know what you're talking about. But you go, number 1, hormone binds the receptor, membrane receptor. That membrane hormone receptor complex signals a G protein. G protein activates adenylate cyclase. Adenylate cyclase turns on protein kinases, activation. Protein kinases activate inactive enzymes, and then they do whatever they're supposed to do. So, just a few steps in there, maybe the major parts, that's a possibility. Let's see, number 3. Oh. I don't know, something with positive or negative feedback. Explain how a negative feedback control system works and give me an example. So, you could say negative feedback, the effects of the hormone interferes or inhibits its own production. An example would be, thyroxine controlling body temperature, goes with a couple of things there. That's a possibility, something with positive and negative feedback. Number 4. Something of the control mechanism, how the hypothalamus controls the pituitary gland, both posterior and anterior. How does it control the posterior pituitary, direct nerve impulses, there's a nerve tract there. How does it control the anterior pituitary, something about the portal system and the releasing and inhibiting hormones would have to be discussed. That's a possibility. What else? Endocrine. If I get bored, I just give you a list of terms and say define the terms relative to the endocrine system. That would be things like upregulation, downregulation, negative feedback, lipid soluble, steroid, stuff like that. So I always throw a few terms sometimes. What else could be a short answer? For the endocrine system. What else? Try to think of now than I think of one thing happening. What else do you think for that? I know there's a few others I asked. I should have put more examples.
Nervous versus the endocrine system?
Maybe, something like that. That'll certainly be in fill in the blanks or multiple choices in some manner. I think I had one on it last time, but it was pretty easy, relative to the nervous system, the endocrine system is, and it had like snow, fast. But yeah, that'd be something like that. Same with, upregulation, downregulation, you kind of need to know what terms mean for defined terms for multiple choice, multiple parts. What else? All right, let me take a break. Let's go to reproductive system. Not as many options there. If I get, kind of a standard boring anatomy one is, for the male, I will give you like a series of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, something like that. And then I'll give you something like, all right, starting at the site of production. Seminiferous tubules. Starting at the site of sperm production is seminiferous tubules. List the structures encountered by the sperm as it moves from the seminiferous tubules to, could be out the penile urethra, to the site of fertilization in the female reproductive system, something like that. All I have to do, I give you blanks, and I give you less blanks than we have terms, but you just fill it out. It would be like, all right, seminiferous tubule. Then you have, what do we have? Straight ducts. Then we had the efferent ducts and epididymis. Then we had vas deferens. It might say include all the glands as well. And if you had vas deferens, then you could have seminal vesicle. Then you could have ejaculatory ducts. Then you have the prostate gland. Then you have the prostatic urethra. Then you have membranous urethra. Then you'd have maybe vulvourethral glands. Then you have penile urethra. And then if go on, then you could have, through the vaginal canal, cervix of the uterus, main body of the uterus, uterine tube, I even list a bunch of things. But just a simple anatomy question. Basically, those are easy because they just have to be in order and fill in all the blanks. And if you miss two of them, it's fractional, so you're talking about 2 points. Pretty easy, but I've done that in the past. Then the other way to do it, keep it simpler, I could ask, or reading like crazy. I could say list the male accessory reproductive glands. For each gland, list its contribution to the sperm as far as semen volume output and any chemical components that are useful or unique or necessary. So obviously seminal vesicles, prostate, vulvourethral glands, and then 60% for the seminal vesicles, 30% for the prostate, 5% for vulvourethral glands, and then just kind of fructose for seminal vesicles, high pH for prostatic secretion, lubricant for vulvourethral glands. So it could be something like that. What else? Let's see, 8. Oh, the other one for reproductive is endocrine control of the female reproductive system. I have asked that in the past. This would be kind of a long answer. So the number of long answers you get depends on how long they're. Some of them are real short, some of them are longer. If they're longer, you get fewer. But I have asked, explain or describe the endocrine control of the female reproductive system. Describe events both in the ovaries and the uterus, and make sure to describe plasma levels of FSH, LH, estrogen, progesterone, and HCG. And I have to go through the whole spiel. That was kind of long, so I tend to avoid it because I'm lazy these days and I don't like to grade those big huge ones. But I have asked it before, so it's always possible. Certainly a piece of it. I would definitely know that endocrine control of the female reproductive system. Because if it's not a short answer, it's going to be a handful of the fill in the blank, multiple choice ones. What else? Development. It'll be fairly light. I can just tell you probably the 2 main short answers for development. List the 3 embryonic germ layers and describe their developmental fate. List the 4 extraembryonic membranes and describe their function in a human. And those are just easy short answers that I think of all the time. They're pretty easy. The only other one, I could have something with slow block of polyspermy, but
What did you say? The germ layers and what they look?
endoderm, ectoderm, mesoderm. Endoderm is digestive system. Ectoderm is skin and nervous system, and mesoderm is pretty much everything else: muscle, skeleton, bone, blood, kidneys, heart. What else? And honestly, you may have one short answer on the industry. I'm sorry, on the development. It won't be big. It's probably the smallest part of the exam. It's still going to the lecture instead. So yeah, from blank, multiple choice, and short answers will be fun. That's all we do Thursday. So when you come in Thursday, just pass out the test and go to work. Ask questions you have. I don't know. Got any more questions for us before we head out of here to lab? Bring that sheet up with you. First thing we do, we get up there. We got a little bit of time. I'll go ahead and open up a cat or two. Point out what you have. Oh, I have posted videos of the cat dissection for blood vessels of the chest and arms. There's another poster of blood vessels in the abdominal cavity. So you've got a couple of sets of videos on Blackboard that you can look at as well, and we'll go over the lab. There's that. See you in a bit.